Welcome to the Quality Assurance and Risk-Based Strategic Monitoring webinar. My name is Cliff, and I'll be your operator today. At this time, all participants are in a listen-only mode. Later, we will conduct question-and-answer sessions. Please note that this conference is being recorded. I would now like to turn the call over to Ms. Tracy Williams. Ms. Williams, you may begin.
Well, welcome, CTN members. We're back here at the CTN webinar series. Today, we'll discuss quality assurance and risk-based strategic monitoring. Elizabeth Alonso, the Director of QA at our Florida node, Maria Campanella, who's the NIDA Clinical Coordinating Center Project Manager, as well as Dr. Robert Lindblad, who is the Medical Monitor at the NIDA Clinical Coordinating Center. They will go into detail about the topic, so I'll turn the discussion over to Elizabeth, Maria, and Bob.
So hi, everybody. This is Bob Lindblad. I'm going to start off today. We'll go through the objectives, and I'll go through the initial slides, and then open that up for questions. And then Maria's going to go through a set of slides, and she'll pause at various spots to open it up for questions. And then Liz will finish up with the last set of slides, and again have an opportunity for questions and answers. So we look forward to people having questions. It's a topic that's received a lot of attention over the past couple of years, and we're really trying to highlight what the CTN approach has been to monitoring within the environment of risk-based monitoring. So the objectives is from this webinar is to really define the risk-based and strategic monitoring, what that means, identify factors to consider when determining how to monitor and how often to monitor, and identify types of monitoring models that would be effective for you depending on what type of clinical trial setup that you have. So in the beginning slides, I'm going to go over the new FDA Guidance Document. That's a risk-based approach for monitoring. And I think this was made available too. It's definitely worth reading if you haven't had an opportunity to read it, if you're involved in clinical trials and actually monitoring clinical trials as well. So the new guidance document was issued in August of 2013. It was issued a couple of years before that as a draft guidance document. They received comments and then they finalized it. And the overarching goal of this plan was to enhance human subject protection and the quality of clinical trial data by focusing sponsor oversight on the most important aspects of study conduct and reporting. And it's sort of interesting, FDA, at the end of the day, is interested in high-quality data. And so for years, the interpretation of that, especially by industry, was to do 100% of source verification. And so that's been what's always been called the industry standard. And FDA has looked at that and looked at those trials and recognized, maybe 30% of a trial's budget is based off of monitoring alone, and that most of that monitoring did not add to the integrity of the data or the trial, and that there are probably better ways to actually look at how things are done within monitoring parameters for trials. So that you get high-quality data. You're not expending your resources just looking at every data element, but really focusing on those areas where there's significant risk or that are pertinent to the endpoints of the trial. So the new guidance document is basically pushing back on industry to say that 100% data verification is really not the way to do it as a rote approach, that they want sponsors to be more proactive, to really think about the risks involved in the trial, think about the participants that are being enrolled and what the data elements are that constitute the primary endpoint in the trial, and to make sure that those things are carefully evaluated during the course of the trial. So it's really moving away from 100% data verification as a rote response to trial management with visits maybe every 2 months or every 6 weeks to a plan that's set up at the onset of the trial to really characterize what are the risks and what's an appropriate level of monitoring for that trial. So FDA, in issuing this, has put forward that they believe that you can improve both the quality and the efficiency of monitoring by implementing a risk-based approach. And again, I think this is an area that's fairly fluid right now within the clinical trial world. If you talk to industry sponsors, they are still doing, for the most part, 100% data verification. Some are starting to move into this area and really trying to think more. This was true in safety reporting, too, where there was new safety guidances and regulations that were put out that are really putting more onus on sponsors to think about what it is they're doing in the world of clinical trials and to use that knowledge that they have to create a more effective program. So the FDA guidance is the rationales that they put forward were really to improve the efficiency and effectiveness of monitoring by focusing on the most critical data elements versus looking at lots of data elements that perhaps are not as critical. One could also make an argument that you might not need to be collecting those data elements as well. And so, in trial setup and trial design, you should make sure that the data elements you're collecting are pertinent to the primary endpoint, but that you're not just adding lots of data elements that have virtually no relevance to the trial and then trying to monitor those data elements as well. It adds a lot of expense to the conduct of the trial and a lot of the burden for the participants and for the trial management team. So you want to try and detect data anomalies in various modes of monitoring, and that can include through the data system or through on-site monitoring as well, too, because you want to ensure the quality and the integrity of the clinical trial data. And to maximize remote data monitoring, FDA recognizes that many trials are run off of web-based central data systems compared to paper systems, and that gives you an ability to do different types of remote monitoring. Some of those are the front-end monitoring where you look at the data elements that are being entered as they're entered and make sure that they make sense in terms of dates or ranges around certain values that you may enter, and really leveraging the electronic data systems as well. You can do back-end checks also to make sure that things make sense relative to other things and send automatic queries out to sites to question why they may do things or put prompts in the data system at the front end to help them collect more high-quality data. So the FDA guidance does not suggest a need for less oversight or less vigilance. I think what they're looking for is really smarter oversight and smarter vigilance that's focused on the areas that make a difference for the clinical trial and that always ensure the safety and well-being of the participants in the trial. So the effort should be focused, which means that you actually have to think. One of the complaints about the industry model is people quit thinking. They just went out and did a very rote, perfunctory review of data elements, looking at what's on the source to what's in the data system, and really didn't think about the conduct of the trial. The FDA is really pushing much harder to have sponsors be actively engaged in the research, to think about the trial and therefore develop a monitoring plan that's commensurate with both the risk and the structure of the trial. It also stresses that the findings that you have should help determine when you take additional action. So a lot of times in sort of the old models, you used to just find discrepancy rates, error rates, and those would get reported out, and basically not much would happen with that unless it was very egregious. They really want monitoring systems to be much more proactive, to work with the sites, to identify problems early, to work on processes so that the systems are actually working as they're set up to do. So the next step I'm going to talk about is the sponsor monitoring within the NIDA CTN, and then after that I'll pause and open it up for questions. So the NIDA Clinical Trials Network, most of you that are on the phone probably know what that is, some may not. It's a platform for conducting multi-site clinical trials targeting substance abuse treatment. The network is composed of academic regional research centers, which we refer to as our nodes, and there are 13 of them currently. And those associated with those nodes are clinical treatment programs, or CTPs, and each node may have, several to numerous CTPs associated with them, so they're affiliated to 140 clinical sites, though only a small subset of that actually participate in the clinical trials. But all the sites are associated with the node within the construct of the CTN, and the node has responsibility for IRB oversight, site management, and training support, in addition to the nodes also having QA activities, and that's where Liz Alonzo is the director of the QA activities for the Miami node. And they do a lot with the sites on a one-on-one basis as well. Besides the node structure, there's a clinical coordinating center that Maria and I are part of, and we're responsible for a sponsor-based quality assurance monitoring program where we go across all the sites, across all the nodes, looking at the activities of each of the clinical trials. We also are responsible for the safety monitoring. We work with protocol development and collection and maintenance of regulatory documents. And then there's the Data and Statistical Center that is responsible for setting up the electronic data capture system, also involved with protocol document development, data quality assurance, and statistical analysis for the studies. So from 2007 to 2010, we had about 4 active trials, 36 sites requiring monitoring, and there was 300 to 1,200 participants in each trial. 2011 to the present, we had 2 active trials, 4 trials that were in closeout, 3 that closed out in quarter 1 and 2 this year, another one that closed out in quarter 3 this year. We had 40-plus sites requiring closeout and monitoring activities, and there are 4 trials currently under development that should be launching within the next several months, which will add new sites, and some of these sites will be fairly research naive as well. So that's sort of the background. I think a couple things to note with that is one of FDA's real pushes is the concept of quality by design, and so they're really looking for sponsors to set up programs that basically are set up appropriately at the front end, and that's the quality aspect, so that it's very difficult for it to get derailed later on in the process, and that you're paying attention to it as you move forward, so you make adjustments as you need to. And this is sort of a metric for businesses as well, too, but I think is relevant for clinical trial setup. So there'll be a lot of things that you'll see in FDA guidance documents and other things that talk about quality by design, and it's really trying to push sponsors to be more proactive, to have better plans, to think about the processes they're setting up. The other thing is sometimes people look at the FDA guidances and say, well, that's really interesting, but we don't do IND trials, we don't submit to the FDA, so I'm not sure why that's important to me. And the FDA guidances are really in parallel with good clinical practice, which is GCP, that's developed through the International Conference of Harmonization, which is an agreement between the U.S., European Union, Japan, Australia. And those are a set of guidances that really outline the conduct of clinical trials on a global basis. And within that are the GCP guidance documents, which all the NIH trials operate under. So the FDA guidances are really in parallel with that and are very pertinent to trials, whether or not they're conducted under IND. Even if it's not under IND, that only means an FDA auditor is not going to come to your site, but it doesn't mean that the trial doesn't have to have the same quality, the same thought process, and the same structure as though it were under an IND as far as safety monitoring and those sorts of activities. So with that, I'm going to open it up for some questions if anybody has any questions, and after that, Maria will speak.
Thank you. We'll now begin the question and answer session. If you have a question, please press star then 1 on your touchtone phone. If you wish to be removed from the queue, please press the pound sign or the hash key. If you are using a speakerphone, you may need to pick up the handset first before pressing the numbers. Once again, if you have a question, please press star, then 1 on your touch-tone phone.
Hi,
I'm showing we have, I'm sorry?
Cliff, this is Tracy. I'd also like the participants to know that you may feel free to use the Q&A feature on the dashboard, and you can type in your questions, and then we'll address them at the various breaks. So that's also an opportunity to get your questions through to the presenter.
I'm sure we have no questions over the phone at this time.
Great.
Okay, so I'm going to turn it over to Maria now, and she will go through our next set of slides.
Hi, thanks, Bob. My name is Maria. As Bob mentioned, I work for the Clinical Coordinating Center. So our involvement in the CTN started approximately 8 years ago. There were some trials ongoing at that time. A lot of trials had already occurred within the CTN. But these were really the trials I'm going to present today, and the approaches that we took then versus sort of the pathway we're forging ahead on now have changed over the years. As Bob also mentioned earlier, a lot of pharmaceutical companies, big models, are still using the current on-site monitoring practice and a more conventional approach. They're typically frequency-based, and they provide a lot of generalized quality control at research sites, but they're usually applied uniformly across all of the sites rather than proportionately to the risks either inherent to that trial or to the site. So I'll go through some of the trials that we've been involved in monitoring and try to give you the perspective of how we've gone from one conventional, that sort of standard model, to one that we would consider looking a little bit more proactively at studies and at the risks associated with them, but also with the sites. We'll start off with clinical trial A. This was a trial that was ongoing at the time that the Clinical Coordinating Center began to visit the sites. At the time, there was an N of 400 participants. That number actually nearly tripled in size, or it did triple in size, based on changes, adaptive changes in the trial. And that was, again, when we had gone through about 50% of the trial. It extended the length of the trial. And there were a large number of participants, a large number of assessments. And at that time, although the trial was using an electronic data capture system, a large number of the assessments were done on paper, requiring paper case report forms, and those did require a heavy amount of source-to-data verification. And that's what the SDV is in there. I should have spelled that out, but that's, for those of you who are not aware, SDV is source data verification. The trial was somewhat complex in that there were 9 sites in various geographic locations around the country, so that accounts for traveling and timing, and specifically making visits became challenging at times because we were taking away from site staff time on site and the principal investigator's time. As I mentioned, there were a large amount of assessments per participant, and a large number of those were paper, and it was also a medication trial, which adds some additional level of scrutiny that a monitor has to adhere to. So at that time, we did take this conventional approach. As I mentioned, it was based on frequency. So we conducted visits to sites quarterly. When I first started monitoring here with the CCC, basically we were assigning monitors to protocols and to sites. And we would basically assign a number of sites, maybe in a range of 8 to 10 sites per monitor, and they would focus almost exclusively on a trial and on their assigned sites. And they did, so they were going repeatedly to the same site. We also looked at 100% of eligibility criteria. That was for all randomized participants into the trials, or enrolled, rather. And we looked at 100% of informed consents, not only the documents themselves, but the paperwork, the progress notes that accompanied that process, and we actually reviewed how that process was being implemented. We looked at 100% of review of primary endpoint data, which are critical data points but we looked at 100% of them, whether we were finding any error rates or not. We looked at 100% of reported serious adverse events. In the participants that we reviewed 100% of all of their data, which are the 10% of the randomized participants at that site, we did review adverse events as well and progress notes for all of those participants. I've got here the word burden in parentheses here because not to say, I want to make it clear that monitoring was not necessarily a burden, but that there was a significant amount of monitoring occurring for this trial. And if you look at the numbers on the bottom, it breaks it into quarters of a year. And you can see that at some points, like in quarter 2 of the last year, rather, that the trial was in implementation, where it was really more towards the follow-up period of the trial, we actually did nearly 3 times as many visits as we did in the beginning of the trial in quarter 2 of that first year, rather. And so at that point, we were really spending a lot of emphasis on catch-up, and you can see that sort of trajectory going from 2006 to 2010. We actually increased the frequency of our visits towards the end in order to comply with the expectations of our monitoring plan, 100% of those critical points. So in 2010, as I said on the last slide, most of these visits were catch-up visits, and we were reviewing a lot of old data. The same visits in quarter 1 and 2 of the last year of the study, we did 67% of the visits were co-monitored. So that means at that point, in order to catch up, we were sending 2 or more monitors to the same site at the same time, which really doubled the cost of each visit, sometimes tripled, and it also prevented us from really looking across the other trials that we were monitoring at that time to try to identify sites that were particularly at risk within that trial, even and on other trials, and because we were still applying everything exactly the same. Obviously, one of the things that changed that had an impact on our monitoring was that change in the number of subjects enrolled, and that, like I said, sort of increased the duration of the study. It resulted in a big increase in costs associated with monitoring that we hadn't previously anticipated. It was hard for us to monitor other trials being simultaneously implemented because we had to push all of our resources in the direction of one trial. It's also an incredible burden to the site staff, who had to have an increased number of visits, and these are multiple-day visits to accommodate multiple monitors. And this was a burden, like I said, not only to the site staff, that includes the investigator, and for the staff at the sites as well as for the staff at the Clinical Coordinating Center, that resulted in a lot of staff morale, a decrease in staff morale, monitor burnout, and a high turnover of staff. So we learned some lessons. All trials have some inherent risks, but each trial, and within each trial, each site, can have varying degrees of risk that require assessment and action that's appropriate for that risk. We realized at the conclusion of this trial that the conventional monitoring plan just wasn't working with the number of assessments and the number of case report forms and the number of visits that we were making. The data errors that we noted, we realized at the end were not timely, and they weren't necessarily always critical to primary endpoint or to safety. Because we didn't identify these problem trends early on, in particular those things that were related to processes, we had a lot of repetitive site staff errors, and these could be things like informed consent, but they could also be errors, breaches in confidentiality potentially. There were other areas that really, had we looked more closely at processes and been perhaps less focused on the source data verification, and the volume of data that we were trying to get through, we might have been able to identify some of these problems early on and address them. And then finally, from the monetary standpoint, the catch-up visits were expensive, and they weren't really a good value. So we realized that we needed to make some changes. Next trial we started in, and then of course we had a lot of trials going on simultaneously, so it's not as though these started, one stopped and the next one started. These were going on concurrently, but we realized with another study that we had going on at that time that maybe we should make some changes and try to be a little bit more adaptive. So we started looking at this risk-based approach. We evaluated and categorized retrospectively the types of trials that we had monitored up until that point. We looked at the number of visits that we conducted and the amount of resources that we utilized to conduct those visits, how much those visits cost, and the impact of the monitoring practices across the trial. We then developed an algorithm, and I was looking earlier at the word risk and thinking to myself, again, not implying necessarily that the trial is risky, but that we want to manage the risk of the trial a little differently. And we started to look in this algorithm at, perhaps the number of sites within the trial, whether or not it was a new or novel approach to an intervention, whether we had research staff at the site who had just been recently hired versus those who had been there for some time and knew the CTN structure. We started to develop a better monitoring plan, and we looked to develop those plans independently for each trial based on that risk assessment. So 3 years ago, we still had the other trial at that point was winding down, but they were going on concurrently. We decided to create a risk-based plan that took some of those things that we'd identified into consideration. So we started to look at what some potential risks associated with that particular trial were. We looked at data collection factors. The previous trial had a significant amount of source documents versus direct data entry. This latter trial had also heavy source documents for some of the assessments, but they were trending more towards direct data entry. We planned up front to adapt the plan during the study based on specific site performance. Again, this is not something we had done with a conventional approach. We were going on a frequency base every 6 to 8 weeks or 8 to 12 weeks without a lot of thought to adaptation. We started to look more closely at how many people had been randomized at a site, how many protocol departures had been reported, the number of adverse events, whether that be high or low. We started to become a little bit more in touch with the risks at a site level and across all of the sites in the protocol. We even looked at things like frequent changes in staffing at the sites to alert us to potential issues with training or retraining that needed to occur. We started to more actively participate in the national team and operational calls so that we could hear the issues that were going on across the sites. We could bring them up to the lead investigators in an operational call, which is a call where the site staff are not as involved, and then we could come up with ways, strategies to address. Did that mean we were going to retrain on a national call? Did that mean that monitors needed to go out more frequently? Did some other action have to take place? So that was a new approach. With trial risk, we looked at the population of the study. What kinds of patients or population participants were we dealing with? How hard was it going to be to recruit them, retain them? We looked at the safety profile of the investigational product. If there was one, if not, if it was a clinical behavioral intervention, rather, then we looked at the safety profile of that as well. How concerned were we about reported safety events? And how much oversight of that were we anticipating was going to be needed? We looked at critical study procedures. Some of the study procedures within our network are complex. As I mentioned, many of them are behavioral, and there are a lot of intricacies that require a certain level of specialization to conduct some of these assessments, and we had to look at each of those individually. We also had to look at any novel outcome or adherence measures. Were there new things that we were implementing, hair sample analysis, for example, or whether we were looking at biomarkers or doing PK assays. These were all some novel adherence measures that we were looking at, and we needed to pay more attention to those processes in particular. We also started to look at the complexity of the protocol design, and this is where it goes back to where Bob was saying earlier. Some of this is really based on designing the trial from the very beginning. How risky are the trials right from the onset? Are they collecting a lot of information in a complex assessment? Do they have a lot of primary endpoints? Do they have a large number of secondary endpoints? What are they trying to accomplish in the trial? Are we experiencing a lot of complexity in regulatory components, looking for perhaps documents that take time to get approval, things that may vary from one IRB to another in terms of what they'll approve, how much documentation is available for review. The sophistication of the EDC system is also something that really worked to our benefit. We could leverage reports and other information that we could get from the data system and from our data management team to help us identify some areas that we could focus on and target on. We also thought it was important at that time to include in our algorithm a way to focus on processes and consistency, and trying to find a way to use our identification of errors as a window into process. So when we started looking at the trials, and when I talked again about risks, it was really considering a multi-site trial might be a higher risk than, obviously, if you're having a study conducted at one site where you have a lot more control. When you're conducting it across multi-site trials, you need a high level of consistency. That is something that is hard to get because there's so much variability with geographic locations, staffing, et cetera. And so it was really important to maintain that consistency. You've got a vulnerable population and you have a high need for confidentiality. The population that the CTN works with in particular, there are oftentimes people who are obviously engaging in illicit activities or who need a certain level of anonymity that may be over and above what another clinical trial might experience. And so that was a risk for us to consider. Varying levels of investigator and site staff experience in research is also important. Same as whether they have investigation experience versus experience within the CTN. The CTN structure in itself, the number of assessments and types of assessments are important as well. Did the trial have a heavy assessment burden for primary outcome, heavy assessment burden on the participant, as well as the site staff? Were there going to be repetitive errors or things missed, misinterpretations of questions? Looking at the pharmacologic therapy, and, again, the novel approaches. Things that might be considered a lower risk in a trial, but not necessarily, again, if it's a novel intervention or a complex intervention, it might be a behavioral intervention, again, when you compare that perhaps to a pharmacologic intervention. Safety profile of the pharmacologic agent that you're using. Is it well known or not? Is this something that's being used for a current indication or for a different indication? Looking at related unexpected serious adverse events. That goes back to safety and pharmacologic agents. But it could also be with behavioral interventions, depending on what that is. What is the risk for instance, a psychological unexpected adverse event? So we started looking at another trial. This is clinical trial B. Very different trial for a lot of reasons. Smaller number of participants, different type of primary endpoint, not a lab value, but more measuring days of absence. There were 10 sites involved in the trial. There were still a large number of assessments conducted per participant, and there was still a large amount of paper source case report forms that required source data verification. The trial went on for about 2.5 years. And during that time, you can see the number of visits that we conducted were a little bit heavier in the beginning and a little bit less towards the end. Part of this is because of a change in the approach that we took to assessing the risk of the trial. We started off monitoring using the conventional plan. We did our quarterly visits and our 100% of 10% of trial participants. It was a relatively low-risk trial. There was the behavioral intervention. There were few safety events reported, no pharmacotherapy involved, nothing particularly invasive, no blood draws, for instance. We had to really identify, was our on-site monitoring identifying any significant issues related to data quality? We weren't finding many. We were repeatedly looking at the lengthy assessments, and we were finding a small number of errors related to the amount of data that was being collected. So we made a decision. As I said before, we had other trials being conducted concurrently. We felt confident in this trial. It was about midway through the trial. The lead investigator was very in touch with the sites, and at that point we made the decision to tailor the plan a little bit more and apply a new sort of risk-based approach where we were actually looking at where we'd come from and where the trial was going, and then allocating our resources based on that risk. So, again, very different types of trials, not comparing the trials necessarily, but more our approach to the trial. We conducted a lot of visits at the end of the trial in a much more reactive manner than we did with Trial B. So that was Trial A, we were much more reactive. Trial B, I'd like to think we're becoming a little bit more proactive in our approach, and that we were looking at aspects of the trial that enabled us to put less of a burden on site staff and on our monitors as well. We limited our ongoing monitoring efforts. We continued to review 100% of informed consent processes, eligibility criteria, and safety events. We also put a primary focus on our endpoint data. We stopped doing the compulsory quarterly visits, and we changed it based on the site-specific triggers. We noticed that there was little to no change in the error rate after the frequency of the visits was decreased. The trial database ultimately locked on time, which is always a good thing, and we concluded that the new strategy maintained the trial integrity, the safety, and the timeliness and morale of the timeliness of the monitoring, and therefore the morale of the monitors and the clinical site staff. Everyone felt at the end the trial had been well monitored and was conducted well. So then we moved on to looking at how could we do the next trial, how could we approach the next trial from our perspective differently, and we started looking at risk. So last year, maybe the year before that, towards the end of that year in 2011 to 2012, that turn point, we decided to look at taking a more tailored approach. Clinical Trial C, a little bit smaller number of participants, 300 participants at 11 sites. They had a somewhat complex plan, but it was also in that we needed to evaluate quite a few data points in order to ascertain the primary endpoint. We started with a more tailored plan, and we identified that clinical trial risk in order to make the plan. We looked at it as a high-risk trial. We had the same substance use disorder population as the other two trials, so that risk was still amongst the higher ones. We were using a novel treatment combination with a pharmacotherapy combination of two drugs, and we were using Schedule II medications, which increases some of the complexity of the regulatory for shipping, for receiving, for prescribing. There was a moderate level of safety risk, but there was also an increased amount of direct data entry, which means that we had less source data verification, which made us uneasy initially, and we wanted to see how those processes were being implemented. And so it was a little bit of a change for us. It made us a little uncomfortable at first. Our monitoring plan, again, we continue to do 100% of informed consent processes, eligibility criteria. We definitely were sticking to the plan of making sure that there was human subject protection in place, that the right people were being randomized or excluded from the trial, and that we were looking at the safety and the critical primary endpoint data. We reviewed all serious adverse events, protocol departures. We reviewed remotely in real time. Those were items that were, as they were entered into the electronic data capture system, our safety monitor was automatically notified of safety events. Our medical monitor and safety monitor reviewed the events in a timely way, responded to sites, requested additional data. We also added the ability for our internal staff to look at protocol departures remotely in real time. This provided another window of opportunity. Instead of the investigator and the coordinating center staff hearing about the departures or learning about them sort of in at the midpoint of the trial when we were prepared to report to a data safety monitoring board. Instead, we were able to see protocol departures as they were reported and start to identify whether trends within a site or trends across the trial, some that at the site level may have required retraining, some across the protocol level that may have required a protocol change of some kind in order to clarify or to avoid those departures in the future, from occurring in the future. We started to review a percentage of the data within the site and then across all sites, and we started to leverage the EDC system. It was always there, but we just started to use it a little bit more to our advantage to identify some trends in reporting. We incorporated data quality features into our plan, and it became a more comprehensive plan. We worked with the Data and Statistical Center to continue to leverage that web-based data capture system. They had already had in place real-time range validation, skip logic, data integrity checks, but this protocol violation review system was built into the system, and again, it allowed for conversations to occur between the lead investigator, the monitors, and the CCC and DSC staff to try to help identify, was this a site issue, a person issue, or a protocol issue. So with clinical trial C, we started out towards the end of the year with that trial. It didn't get started until a little bit later in the year. And by the time it ramped up and was fully sort of in swing, it was 2012. And we did the bulk of our monitoring in that time period, in the 2012 year. And in fact, most of the monitoring occurred in that third quarter. We noticed that this really gave us the ability to focus on certain aspects of the trial while it was in active implementation phase, and to transition towards less monitoring of those types of activities, more in terms of data cleanup, helping site staff to resolve safety events that were ongoing, helping to wrap up the medication and return to the pharmacy. So it was a very efficient approach to monitoring, and it really did help us to move the trial along quickly so that we could close and lock the database in a timely fashion. This information here is another way of looking at that same information. You can see the same number of visits conducted in each quarter, but the difference is that we also started to incorporate the use of remote data review. And so, as I mentioned, there are some lengthy assessments that are conducted within the CTN that are very heavy on source documents. And it's largely because it's based on participant self-report, and some of the trials, if you have a trial that's a year in length, for instance, you could be asking somebody for this self-report information, which you'll see there is TLSB. It stands for Timeline Follow-Back. It's a lot of assessment going on. It takes an incredible amount of resources and valuable time to have a monitor review, but it's oftentimes linked to primary outcome. So it's important data to be reviewed, and recognizing that, though, we didn't want to spend a monitor's valuable time where they can actually see processes in action at the site level. We didn't want to spend the time doing that while we were on site. We recognized that we were dealing with information that was de-identified, that we could collect here at the Clinical Coordinating Center and have someone here review that information. Source data verification, they're comparing one field to another for a transcription error to make sure that the integrity of that data, that we felt that it was valid. But then we could also spend a good chunk of our time doing that towards the end of the trial. Well, we did some at the end of 2012, and then we did it again in 2013. We did sort of two sweeps of remote monitoring where we were actually able to conduct a large number of those reviews here in-house, which meant less burden on the site staff. There was still some burden on the site staff because they did have to send the documentation to us, so it was not without burden completely. But it definitely didn't involve identifying a date and a time that we had to have all staff there present to discuss these case report forms with, because we could do it here and then get back to them with a report. We did issue reports for those reviews, so the sites were receiving fairly instantaneous feedback on the reviews, allowed to revisit some of their processes if necessary. and it also allowed us to increase the level of monitoring that we were conducting for that variable. We realized that it was appropriate to tailor the sampling rate and type of visits conducted, so whether we were doing on-site versus remote, according to the phase of the trial. So, as I mentioned, we did some of the heavier monitoring in 2012. They were on-site visits targeted towards that active phase of the trial when we were most concerned about participant safety because people were on drugs at that time. We also looked at the follow-up phases a little differently. Instead of going so much on-site at that point, we were doing regulatory reviews here in-house because we have an electronic system that is also web-based that allows sites to upload regulatory documents, copies of the regulatory files to us for review. So there were a lot of things that we were starting to recognize we could do more efficiently remotely with less burden on site staff and less burden on our staff. The rate of the visits appears at this point to have been appropriate, rather, for the clinical trial that we implemented this tailored approach to. We were able to identify issues and assess them throughout the trial. We had increased communication with the lead investigative team where we actually discussed trends, concerns, and how we could take more targeted approaches to monitoring both within the site and within the trial. We definitely took a more proactive approach versus a reactive approach that we did in clinical trial A. We recommended basically additional data quality checks based on real-time monitoring findings, and we reviewed these PVs remotely in real time, which again provided us great windows opportunities to look into processes, and it allowed us to address the issues quickly. So looking ahead, I think this would probably be a good chance for me to take a minute to ask if there are any questions.
As a reminder, if you wish to ask a question, press star then 1 on your touch-tone phone. And I'm showing we have no questions at this time.
Okay, great. Thanks, Cliff. Okay, so looking ahead, I've kind of gone through a little bit about sort of how our epiphany and how we came to realize that, and this is when the guidance document, this is before the guidance document came out from the FDA 7 years ago, and then sort of we got some affirmation about the plan when the guidance draft came out a few years ago. We thought, okay, we're moving in the right direction. Industry is going to catch up eventually, and we decided, again, it was affirmation that we were going in the right direction. So looking ahead, we've learned a lot in the last 7 years, and we're thinking a lot more critically from the very beginning. When we first get a protocol concept, we're starting to think already about what kinds of things are we going to be concerned about that are risks in the trial that we can help to mitigate. So looking ahead, thinking about being proactive, we started to use the same criteria that we've applied to clinical trial C, which is where we're using that more tailored and adaptive approach. We started off by looking specifically at the risks of that trial, specifically at the risks within each site, and then we decided that we could adapt along the way based on the phase of the trial and what was happening, the issues that we identified throughout the conduct of the trial. We'll determine some critical variables associated with participant safety primarily, and then with the outcome and overall trial integrity. We're going to look for critical elements also that are at risk for errors, either in interpretation, again, site staff who may be interpreting a question within a site a certain way, answering the response, or it could be a participant that's answering a response to something that they believe is the correct question that they're being asked, and it could in fact be very different from the intent of the question or the trial. And we also would look at processes like informed consent or these lengthy study assessments or medication dispensing. So we'd start off critically, identifying those elements. further enhancing the plan by conducting the on-site visit as soon as the first participant is randomized. We were a little slow to catch on to that. We knew it had to happen, but it happened less quickly than I think we should have done it. Looking back now, and I think we've learned as we're moving along that we need to hit the sites fast and hard to begin with to make sure that these processes are moving in the direction that they're being implemented in the way that it was intended for the trial. Concluded that probably more frequent visits in the earlier stages of implementation, and then reassessing on a site-by-site basis instead of applying that frequency model. We'd adapt the overall plan with every protocol amendment as necessary. So instead of having one monitoring plan that sort of sticks through the life of the trial, if you've got a trial that goes on for 3 years and it goes through various phases and various adaptations, whether it be a change to the number of participants enrolled, dropping or adding of sites, a variety of high staff turnover, any of time that there's any major change, either with the protocol or the implementation of that protocol, then we have to adapt our plan accordingly. We also have to consider adapting the plan per site necessary. There are a lot of benefits associated with risk-based monitoring or this tailored or strategic approach to monitoring. It allows us to definitely use our monitoring resources a little bit more wisely, more efficiently, and more thoughtfully. Reviewing ongoing trials, so far we haven't identified any problems with data quality or monitoring frequency. Taking proactive approaches seem to be identifying trends in data earlier, and they're allowing us opportunities for early corrective action, and it allows us to improve processes. There are some potential risks, however. You have to change your expectations a little bit. I think sometimes some of the assumptions that we made early on about a trial and what those associated risks were have to change. And you may identify throughout the course of the trial that something that you thought was not going to be an issue because you thought this is a no-brainer, everybody's going to get this right. There are risks, and people will implement things in a different way, and mistakes will be made. So I think assumptions about the trial are going to have to change throughout the life of the trial. There are also protocol-specific training requirements. If you're going to have fewer visits, when we had the more conventional model, we actually had the monitors going on site, conducting a lot of on-site training. We're going to actually expect that we've got highly trained study teams, highly trained site staff that are getting constant reinforcement and retraining. Sometimes, if you think about if you don't have sufficient and timely training, if you don't have up-to-date manuals of procedures, very clear SOPs at the site, very clear lines of communication between the principal investigator and the site staff, you've got a lot of risk for not catching these things if you're not going to sites, on-site monitoring as often. As Bob had alluded to earlier, the FDA guidance is not suggesting that you're less vigilant or that you do less monitoring. What it's suggesting is just that you do smarter monitoring and more strategic.
So one thing I was going to add to that was that you can take away a message from this that says, okay, we just have to go to sites less and everything will be okay. We actually work harder doing this. It's harder to schedule monitors. It was easy when it was quarterly. Everybody mapped things out for a year. People just showed up at a site, did whatever they could do there. Maybe no cases were enrolled at that site in that interval time period, and they'd still go and sit there and look at whatever they'd look at. So for us, it's a lot more work on our end to make this work and make it work to maintain quality. So I don't want anybody to think that A, it's easy to do, or B, that you don't have to put a lot of time, thought, and effort into it, and that the result of this is you just go to sites less frequently. Some sites you may actually go to more frequently than you would have otherwise, and some less frequently, and you have to be adaptive to deal with that through the course of the trial. You have to pay attention. You have to think about it. So again, I don't want the wrong impression to come out that it's just decreasing monitoring visits. It's actually a much more proactive, thoughtful, and in a lot of ways time-consuming process to make this work and make it work well.
So real briefly, I just wanted to touch on the fact, thank you, Bob, that there are many monitoring models. No one absolutely is the best. You are trying to cover as much ground as you possibly can. Our jobs are to ensure that participants are safe and that they are protected, and then that the integrity of the trial is worthy of the expense of the trial and the investment of people and resources in the trial, and that what you get out of that trial is something conclusive that will help you to move on beyond that trial. So different monitoring models are going to work for different trials, and they're going to work for different types of structures. So one of the challenges is to come up with a model that works for you. We are fortunate in the CTN that we're able to have a flexible model somewhat, and we'll go through that a little bit more with Liz's presentation in just a few minutes. ICH E6 and ISO guidelines both advise sponsors to assess and consider the objective, design, complexity, size, and endpoints of their trials. And this goes back to Bob speaking about quality by design, starting with protocol development and then going all the way through, continuing to train people and to be consistent in the message of the trial. When you're choosing a model for your monitoring and determining whether or not to implement a more strategic approach, you do want to take into mind things like the number of sites and the number of participants in the trial, how long the trial is, so you can target active phases versus follow-up phases. You do have to take into consideration the experience of your research staff and the complexity of interventions and assessments. Another thing that we really didn't discuss too much here, but touched on it briefly, was the idea of remote monitoring. There are many remote activities that can happen, and oftentimes they're lumped together into a term of centralized monitoring. Centralized can mean the data system, but it can also mean, in other words, having those front-end checks and integrity checks that were mentioned earlier, but you can also have data managers looking at the number of errors, the frequency of errors, and then we've got people that can look at remote reviews of regulatory documentation. Some companies are even moving towards using encrypted informed consent forms. I read something earlier today that talked about that practice, and it sounds surprising, but finding ways to review those documents in real time so that you feel assured that no one was enrolled into the study that wasn't eligible and that didn't sign consent. So there are a lot of ways you can do some centralized. Again, the emphasis is not so much that you do one versus the other, it's just what you do one versus the other, so how you spend your time on site. Centralized monitoring can be efficient and cost-effective. It can allow for some target and random selection of data. And you can see trends both within sites and across the sites, which is also important. You can look at things like audit trails. Who's entering the data? What time are they entering the data? How often is that same person making the same mistake? Those are important things to trend within the site and then across the site. But on site, you get a better window into processes. Those are things you can't see through centralized monitoring necessarily. You want to look at these informed consents. You want to look at how labs are being delivered, how faxes are being received, how patient information is being kept confidential or not. You can verify a level of PI oversight, looking at levels of staffing. Is one person doing 6 jobs? You can look at site logistics. Is one research site 6 miles down the road from their pharmacy? How does that work? You're also providing some reassurance that someone is personally watching the site. When you've got centralized monitoring, there's a big detachment from the sites to the oversight, and I think it is important to have people continuing to go to the site frequently enough that sites feel some accountability and some investment in the research, and that they're part of a larger team. You're also looking for site staff compliance with site SOPs and with IRB expectations. So our recommendation would be to take advantage of the option to do both. Identify the things that have to be done on site, so the ICF reviews or medication accountability, but then identify the other things that could be done remotely. What kinds of assessments, for instance, the one I mentioned that we reviewed remotely, what kinds of internal audit checks can you do? So I can open it up one more time to questions, and then I'll turn it over to Liz.
Once again, if you have a question, press star then 1 on your touch-tone phone. And I'm not showing anyone queued up.
Okay, Cliff, thanks. And actually, I spoke a little too soon. Liz, I'm going to take a few more slides from you before I turn over to you. Thanks.
Fine.
So, as I mentioned, the CTN, we're lucky within the CTN because we actually do have some flexibility in that we have a structure that allows us to have the eyes of a data management center, a data manager. We have the eyes of a protocol specialist here at the Clinical Coordinating Center, our sponsor contracted monitors who go out to the sites, who also review data internally. And then we have this other level of oversight that is the node, and the node that is sort of facilitating and training and offering support and guidance to the research sites. And so that's where we're able, within our network, to leverage the role of monitoring. We've got, as I said, the CCC, we've got the node, we've got the Data and Statistics Center, and then the one that I didn't mention was the site itself. This is a really, if you look at where it all starts, it starts at the site. And so how well that site staff is trained, how well that they are starting collecting data, entering the data, documenting what they see and hear. These are all where it starts. We're coming behind all of that, but it has to start at the site level. Otherwise, we'd have nothing to do. Learning to manage that risk through monitoring, looking from the different perspectives of the Clinical Coordinating Center monitors versus the local node monitors, the sponsor monitors are able to visit all sites. We do on-site visits to all sites across the entire protocol. It gives us a different view of protocol implementation. We're looking at it for consistency across sites, and we're really very concerned with, is everybody doing this in the same way or as much in the same way as possible? And if not, do they have some operational procedures in place that will account for why they're doing things a little bit differently? We're able to implement risk-based and adaptive monitoring and centralized reviews, and we're able to do that fairly easily. We also have experience with CTN trials, which is helpful with a variety of CTN trials. So we can oftentimes identify issues that happen in one trial and maybe apply those risk assessments to another. The node monitors are the ones that are closest technically, not necessarily in geographic proximity, but often and ideally in geographic proximity. They have fairly, easy access to the sites. They've got a good relationship with the sites. Oftentimes they've either hired the staff, definitely trained the staff. They have extensive experience with CTN trials. They've either been lead investigators themselves or they've participated in numerous trials over the years. The network is nearly 15 years old, and it definitely, these guys have a lot of experience with the trials. They're knowledgeable about their local site practices. If they're in the same state, it definitely helps, but even if they're not, they know the state laws, they know the state SOPs, they have the relationship with the IRB, so they're the ones that can help alleviate those responsibilities from the site staff or for the site staff, and that gives them a different type of a relationship. Okay, so with that, I guess, segue, I'm going to turn it over to Liz to wrap this up.
Great. Thank you, Maria. So we're going to shift the focus a little bit now. As Maria was discussing, there are different levels of monitoring and oversight that occur in the NIDA Clinical Trials Network. And just to put this in context, Nodes are structured in different ways, and management and oversight responsibilities can be distributed also in different ways. So at some nodes, you might find a protocol manager model, which is one individual that takes on both monitoring and oversight and also coordination responsibilities at the node level for a particular trial. Or at other nodes, these responsibilities can be distributed between two individuals, with one person taking more of the monitoring role and another taking more of the coordination role. And the person in the coordination role is sometimes referred to as a node project coordinator or a node study coordinator. So I'll be referring to sort of the umbrella of all of these local management and oversight responsibilities. So I'll use the term protocol manager to refer to, as I said, to all these responsibilities together, the management and the monitoring responsibilities together, which can be conducted by one person or by two different people, depending on the node. Okay, so let's talk about the role of this protocol manager. As we mentioned, the CCC, the Clinical Coordinating Center, really works on behalf of the sponsor, and they are the sponsor's appointed monitor, whereas the protocol manager works on behalf of the node study PI and site PI. So as we all know from our GCP training, we all share the responsibility. We share the responsibility for the protection of human subjects. We share the responsibility for regulatory compliance and for collecting quality data. And so as part of meeting that responsibility, it's important that at the node level, we do have oversight, direct oversight of the sites that are conducting the trial as part of our node. Okay? And the protocol manager really plays a key role in helping the node to meet that oversight responsibility. Okay. So the protocol manager works closely with the study site and therefore has a unique perspective. They often serve as a liaison between the node or the university and the study site team, and are able to visit the site more frequently than the CCC monitor. In some cases, they might be in the same city as the site, same state. In other cases, they may not, and visits may also be remote in this case. But there tends to be a lot more contact, participation in study meetings by phone, and more frequent visits. Okay. The protocol manager may be the person responsible for regulatory submissions for that trial at the node, so corresponding with a local IRB. And if they're not the person directly responsible for that, they can definitely serve in a very supportive role as far as tracking that all safety events and protocol deviations and such are reported to the local IRB, and that the study is conducted locally in compliance with local IRB policies. Okay. The local monitor or protocol manager tends to be very familiar with local personnel and procedures at the site. So because the protocol manager has this unique perspective and a close relationship with the site, they can really help to fill in the gaps in terms of providing close monitoring and support focused on local processes and procedures.
Liz, this is Maria. I want to interrupt you for just 1 second and ask if maybe you could give us a few examples of something that may be a gap.
Sure, definitely. I actually will be covering a few of those in the subsequent slides, and I'll add a few more. So a number of the trials in the CTN involve socio-behavioral interventions, for example. And so this is one area where the CC monitors tend to do less oversight or have less involvement in. And it's an area where local monitors can really be of great support. So for example, reviewing progress notes for the study intervention is something that can be done by the local monitor. This is important for a couple of reasons. Number one, we want to make sure that the study therapists or interventionists are complying with the documentation requirements for the intervention. But also reviewing the intervention progress notes for safety events is a very important activity. There may be safety events that are recorded in the progress notes that are not identified as such but would qualify for reporting based on the trial's definitions of safety events. So that's one example of an important contribution that the local monitors can make. Also, helping the site or supporting the site with any recruitment or retention challenges. If the site is having difficulty meeting their recruitment target or bringing patients back for follow-up visits, because of this close relationship, the local monitor can help to really look at a micro level, to look at what's going on with specific participants, what's the level of outreach, of contact, the number of attempts that are being made to bring a participant back for a follow-up visit. Is the site using all of the different strategies that could be used to sort of re-engage a participant who's not being responsive or, coming in for a follow-up visit? So these are a couple of the areas. There are numerous where a local monitor could be of support. Okay. Does that help, Maria?
Yes, thank you. That helps a lot. Thank you.
Sure. So, and I'm going to continue to talk a little bit more about how the local monitor can really be of support to the site. Great. So some of the areas where the local monitor can be very helpful are training and retraining of staff. So we have these very large protocol-specific trainings at the beginning of the study, which are very helpful. But typically there is the need for additional training and support throughout the course of the protocol. It's very common for there to be amendments to the protocol changes to procedures, and it's important to provide training and support that's timely and responsive, as far as keeping up with all of these changes. The site team is very focused on doing their job, recruiting the participants, collecting the data, and so it's very helpful if they have somebody at the node level that is supporting them in this process, that's keeping track of all of the changes in the protocol, in the procedures along with them, and helping them to make sure that they are adhering to these changes and receiving any additional training that they may need to then comply with these changes in procedures. There can be a very high rate of turnover sometimes at study sites, and so in the case of turnover, the local monitor or protocol manager can assist with the training of new staff to try to minimize any gaps in coverage, and also just making sure that there is proper documentation of staff responsibilities, qualifications, and staff training. Okay. The protocol manager can also be very supportive in the process of developing and maintaining local SOPs. So as I mentioned, oftentimes there's changes in the protocol or in the procedures throughout the course of the trial, and these may also call for changes to local SOPs. So the local monitor can assist in that process. And in really just taking a look at the procedures that are occurring locally, how are responsibilities being divided among the different staff members on the site? How is the site working with the study laboratory or other sort of collateral services or offices that the site may need to cooperate with in carrying out the study, and helping to examine these processes and streamline them and make them more efficient where possible. Okay. So another area where the protocol manager can be helpful is in the prompt identification of protocol deviations or violations, making sure that they're documented appropriately and reported here in the EDC as appropriate, and also to the local IRB, and helping the site to institute corrective and preventive action plans. So trying to get to the root cause of why did this deviation occur. Was it staff error? Is there a procedure that's vague that needs to be specified further? Does there need to be a change in a local SOP? Is there some sort of misunderstanding with regard to a specific study procedure or a specific form? And really helping the site to get to the root of whatever if there's a larger issue or trend, getting to the root of what is the cause of that problem and addressing it to try to prevent future errors. And in the area of safety events, the protocol manager can definitely assist with identification of these events, as I mentioned, in reviewing progress notes for study visits and for the intervention. The protocol manager can identify whether there's safety events that have not been documented and reported, and also just helping with that process of documentation and reporting to the IRB. Regulatory compliance is another area where the protocol manager can be of support. Again, because of that close relationship with the site, and work with the site to make sure that updates are made to the regulatory binder promptly, that protocol departures and safety events are reported to the local IRB. And as I mentioned, also providing support in the preparation of IRB submissions, whether it's the initial submissions, amendments, and continuation reports. Okay, as far as data review, one of the ways in which the protocol manager can help is by reviewing data reports. So there are a number of different reports that can be generated directly from the EDC that will help you to recognize anomalies in the data, missing data, values that are out of range. You can generate these in real time, and it can really assist in the process of helping the site to make sure that their data is as complete as possible. Okay, when it comes to data, it also, again, we're talking here not necessarily about monitoring less within the risk-based approach, but also monitoring differently and monitoring more efficiently. So having the local monitor then focus to a greater extent on forms or processes which are not reviewed by the Clinical Coordinating Center and others. And an example of that, as I mentioned earlier, is documentation related to the study intervention. The protocol manager can also review checklists and progress notes. So here you can get a sense of what's going on more at the micro level. Are there certain challenges that are occurring in completing study visits? Are study visits having to be interrupted and carried out over multiple days due to medical procedures or different things? So there's a lot that the monitor can really, and the local monitor can learn from reviewing those progress notes, contact logs with participants, and work with the sites on any local issue. Okay. And then looking at quality and quality assurance and monitoring on a continuum, as Bob and Maria mentioned earlier, really where it all begins is with the site and internal QA practices. Okay, so quality assurance begins with internal monitoring and the expectation of a high standard of data quality and regulatory and GCP compliance. And we like to refer to this as a culture of quality, where really it's all about a commitment to doing good work, a commitment to our participants, to protecting their safety and their well-being, and to carrying out good research and collecting high-quality data. Okay. So there's specific strategies that study sites can carry out as far as just increasing, I guess, the quality of their procedures and their activities. One area where local, say, internal quality assurance can have a very big impact is in the area of informed consent. There tends to be, I would say this is probably the area where in CTN trials we've had the greatest number of deviations and violations. And so a real-time review of the completed informed consent can really go a long way as far as reducing errors. And so the recommendation is that as soon as you've completed the informed consent process with a participant and they've signed on the dotted line and so on, that you take a moment while you are still with the participant and review everything on the consent form. You want to ask yourself the question, are all printed names, signatures, dates, and in and out responses present and correct? This is it's really an invaluable opportunity while you're still with the participant. It takes a few moments, but it really allows for prompt corrections and reduces the need for reconsent, and can greatly reduce the number of protocol violations. If there's sufficient staff available, the form can be cross-checked by another staff member or just double-checked by the same staff member. Okay. Another area where internal QA can go a long way is in the accuracy of data entry or abstractions. So fortunately, we are able to do a lot of direct entry now, but there are still some forms or some areas of the trials where we're still entering data from a paper source document. And so this calls for a great level of just attention in making sure that we're entering the data correctly. For example, on some trials we are printing lab results from the electronic medical record and entering them into the EDC. So doing a cross-check or a double-check that these values have been entered correctly can really help to reduce errors in that area. Okay. And then, as I mentioned earlier, regular review of data reports, such as missing forms, missing values, integrity data queries, and such can help to get a real-time snapshot of any issues with the data so that you can correct that as soon as possible. And really the spirit of internal QA is taking a very proactive approach to minimizing errors, identifying what are those errors where, what are those areas where errors are likely to happen, what are the more sort of complex study procedures, and then implementing very targeted strategies to try to increase accuracy and reduce errors in those areas. So one example is more and more we're using tablets for data entry in trials. And so using a stylus pen in conjunction with the tablet can really help to increase accuracy in data entry. It's just a small change, but it can really go a long way in terms of greater accuracy. There are many different things, just to provide some examples. Distinguishing ICF versions can sometimes create some confusion. We have revisions in our IRB approved consent forms. And so I've seen some sites use different color papers to identify different versions to make sure that when they're shifting from one version to the other, there's no confusion with using older forms. Okay. Also the use of checklists to sort of break down and make sure that all of the steps in a complex process are being carried out appropriately. And the use of reference or guidance documents displayed in key locations. For example, the steps for processing a urine test can be displayed in a lab area where the tests are being processed, just as a reminder. Okay. All right, so I think we'll break for questions now.
And as a reminder, if you wish to ask a question, press star then 1 on your touch-tone phone. We do have a question from Paul Joachim. You may go ahead.
Hi, this is Paul Joachim. I have a question. I mean, so in terms of the local monitoring, well, then that means it's going to vary from site to site. Do you all share success stories during protocol conference calls, weekly or whatever, in terms of sharing lessons learned or success stories in terms of monitoring, local monitoring at sites?
Mm-hmm. Hi, Paul. This is Liz. So I can speak from my own experience in the trials that we've led in the Florida node. We have local quality assurance calls where all of the local monitors come together periodically, and we do share trends that we're finding at sites, specific issues and stories, anecdotes about how we were able to support a site with a specific challenge or reduce areas in a certain part of the trial. So that's something that we have done to help ensure some uniformity, and also just so that the monitors can be of support to each other and, like you said, share ideas and strategies that may have worked at one site that might be helpful to another site. And typically, the feedback from the monitors is that they do find those calls helpful, and just to hear the experience of other monitors on the same trial.
Great, thank you.
Sure.
I'm sure we have no further questions at this time.
Okay, great. So we will go ahead and wrap up with the factors to trigger monitoring actions. Maria?
Hi, okay. So we'll wrap it up with just, again, going through some of the very basic things that we've talked about throughout the presentation to really help you to determine what type of approach to monitoring you're going to take, whether it's from a protocol standpoint, a site standpoint, whether you're coming in as a node protocol manager at that site, looking at the complexity and length of a trial, the implementation of the plan of beginning your monitoring sort of fast and hard. You're going out, and as soon as the first participant's randomized, you're really going in and looking at detail at processes. At that point, you're not as concerned about data as you are about processes. You're looking at the volume of source-to-data verification. How much documentation are you going to be dealing with? When we go out on a monitoring visit, we definitely look at how much data has been accumulated up until that point, so we can decide what the most appropriate how to triage the visit, how to place the most emphasis on the items that are most important for us to get through at that visit. You definitely want to look at how many sites within your trial, or whether the site within your node is relatively either naive to research or to the CTN. Again, the structure is a little different, the types of calls, the way that the network works, maybe the assessments. You want to definitely use performance indicators, and Liz mentioned reports that are available in the trial progress report that are available to lead investigators, site investigators. They're available to QA and to MS and even to the sponsor, that really help to determine. They're looking at how many forms haven't been completed within the 30 days that they're expected to be completed, how many forms are not being completed at all. And where are there a lot of missing data. So you can look at even protocol deviations or departures. Those are things that are performance indicators, rates of randomization, that those reports can really be helpful in triggering more monitoring or more oversight. You can start to look at the number of error rates identified from remote monitoring, and you can just do some good due diligence. I think that's pretty much going to wrap up our portion of the presentation. Tracy, I know you have a few more things that you wanted to say. We can open it up one more time to questions.
And as another reminder, if you wish to ask a question, press star then 1 on your touch-tone phone. And I'm showing we have no questions at this time.
So everyone, thank you so much for attending. And I know as you're thinking about varying your monitoring models, we will make this presentation available on the CTN Dissemination Library's website, so you can refer to the information as you're going through the changes and development procedures. Maria, Liz, and Bob, thank you so much for this information. It's sort of a refresher, but also takes it a bit further by going into some of the nuances that we're taking the monitoring for the CTN and sharing it with others. So thank you very much.
Great.
I remind participants to also take the survey that we will be sending out to you via email, and let us know how you enjoyed or what suggestions you have for improvement for our future presentations, and what types of topics you would like to either discuss as subtopics to this one or some other new topics. And we invite you next month to our presentation on adaptive research design for substance abuse clinical trials. We'll be sending out invites via email as well.
Tracy?
Yes, Bob.
One other thing I'd say is if any of the people that are listening on this have questions for us, or maybe setting up a monitoring plan for a trial or even a multi-center trial, and they want to run some ideas past any of us, that our email addresses are available. Feel free to send an email and just bounce off some ideas. It is a thought process. It is iterative, and we're happy to share any information we have that would make it easier for anybody listening in.
That's a good idea, Bob. One central place folks can reach out to is ctntraining@ms.com, and I'll definitely get you in touch with, particularly Liz is a resource on the local level, but as the NIDA Clinical Coordinating Center, we have Bob and Maria and others who can work with you. So thank you all, and this will end our webinar. Thank you.
Thank you, ladies and gentlemen. This concludes today's conference. Thank you for participating. You may now disconnect.